We're going to move to our last speaker. Her name is Choi, and she's actually going to talk about the, I think, trophallaxis behavior that was just mentioned at Jean Robinson's lab.
Hello, can you hear me and see the slides?
Yes, yeah, yes. We can hear you.
Yeah. Okay. So these two honeybees are interacting with each other by sharing food from mouse to mouse. And this is called trophallaxis, as you heard from the last talk. And my experimental collaborators, the Jean Robinson group at UIUC, can track each of these trophallaxis events by putting a barcode with unique IDs on each of the honeybees. And so that we can track each individual honeybees and study their social interaction. There are thousands of honeybees in each colony, which means that we can get lots of data from each measurement. And the simplest quantity we can look at to study their social interaction is the duration of the trophallaxis. So my collaborators put the honeybees in this two-dimensional observation hive and tracked their interaction using the high-resolution camera shown here. And if we plot the distribution of the trophallaxis duration or the interaction time in log-log scale, shown here, you can see that for all the colonies we measured, the interaction time distribution is power law distributed. Obviously, biological systems are very complex, but we wanted to come up with the minimum model to explain why you would see a power law interaction time distribution. To do that, I viewed the bond between a pair as a particle with two states representing an interacting pair and non-interacting pair. And the particle changes each state by jumping over the energy barrier. And so the time it takes for this particle to escape from this potential well is the duration of interaction or interaction time. For a single barrier height case, this interaction time distribution is exponentially distributed, which can be calculated by calculating the probability not to jump until time t and multiply it by the jumping rate omega. If the pair has a large energy barrier, it is more difficult for the particle to jump over the barrier, which means that this pair will tend to interact longer. So this variability in the population can be represented by the distribution of energy barriers, P of E. And we can guess what P of E would be by noting that the B pair with the highest energy barrier is the stablest. So let's say this center bee wants to interact with one of the honeybees in its neighborhood, and it will end up interacting with a honeybee with the largest energy barrier because they will form the stablest pair. So we deduce that P of E would be the extreme value distribution for maxima, especially the Fisher-Tippett-Gumbel distribution shown here. We are only interested in the heavy tail behavior at the large T. So we take the large energy barrier height limit and end up with an exponential P of E. Now we have exponential pair interaction time distribution and exponential energy barrier distribution. By using the Kramer's rate formula shown here, we can calculate the population interaction time distribution F of t, which turns out to be a power law at long times. And this is actually what we observe in the experiment. Note that the exponent in the F of t is related to the exponent in the P of E. So this exponential barrier height distribution is what the theory predicts, but the energy barrier is not something we can measure directly. So by using the Kramer's rate formula again, we can transform this P of E into P of tau, where tau is the mean pair interaction time. And P of tau is actually a power law with the same exponent as F of t. So by comparing the scaling behavior of P of tau and F of t, we can verify this prediction and thus verify our theory. Before I go further, I'd like to be extra clear about this f of t and p of tau. So t, which is the population interaction time, is the duration of each interaction event. So we have 3 honeybee pairs in this case, and each honeybee pair interact multiple times, and each interaction event has this duration t. So we collect the duration of each interaction event for all pairs and get the distribution f of t. On the other hand, the mean pair interaction time tau is the mean value of the interaction times between a pair. So we get tau for each of the pair and collect them from all the pairs and then get the distribution p of tau. And if we plot f of t and p of tau in log scale as before, and we'll see that indeed they have the same tail behavior with the same exponent for large time. And I'm showing this result for only one colony, but we see the same result, basically the same plot for all the colonies we analyzed. What's very nice about our theory is that it applies not only to honeybees, but also to other systems such as humans. So we got the human face-to-face interaction data from social parents collaboration for various different settings, such as schools, hospital, conference, etc. And for all these different settings, we see a power distribution of the interaction times for humans, just like honeybees. And we compared the scaling between f of t and p of tau for humans as well. And you can see that indeed they show the same scaling behavior. And here again, I'm only showing the data, only one of the data sets, but we see the same tail behavior for all the data sets we analyzed. So what our theory tells us is that this power distribution, as well as the scaling law, is due to the individual differences in the population. However, only recently, the individual differences in worker honeybees have started to be recognized. So we had to directly show that the worker honeybees are actually different to convince the people in the field. So we measured the Gini coefficients for total time spent for interaction, total number of interactions, and total number of partners for all the data sets we analyzed. Here, Gini coefficients is the quantity used in economics to quantify the inequality in the society. So in our case, it means the inequality in the individual contribution to the social interactions in the community. And here is the result. So I'm only showing the result for the total time spent for interaction, but the result looks the same for other criteria that we saw. So if the society was completely equal, the data points would lie on this linear reference line. And the more unequal the society is, the more deviation you will see from this reference line. And you can see that for both honeybees and humans, they are both unequal societies. And we calculated the actual value of Gini coefficients. And the closer the Gini coefficients is to 1, the more unequal the society is. And by looking at both these plots and the actual value of Gini coefficients, we can see that although honeybees are unequal society, they are less unequal than the humans. And we speculate that it could be due to the closer genetic relatedness of the honeybee colony, because in our experiment, the queen bee was inseminated with only one male. So the sisters share about 75% of the genes. So, so far I showed you that for both honeybees and humans, the interaction time distribution is heavy-tailed. And this heavy tail and the scaling law we saw in our theory arises from the individual differences in the population, which could be represented by the energy barriers. And we also directly showed that there are indeed individual differences among worker honeybees. Yeah, thank you.
Thank you so much.
Thank you.
Let's see if we have any questions. Yeah, we have one from Anton.
Antonio is here.
So,
Can I just
Yes.
actually yeah. So you mentioned that there's individual differences between the honeybees. I'm wondering whether I can think of this as an out of equilibrium process where basically the energy barriers are kind of like, instead of changing over time, they're changing over individuals. Is that kind of the picture?
So you mean
Because
the energy barrier changes over time?
the same bee over time changes the interaction properties, I'm guessing.
So we didn't consider such a possibility, and it seems that even if we don't consider such such feature, the data agrees with our theory. So probably even if there is fluctuation, I think the fluctuation is small enough to be able to assume that, safely assume that it is a constant.
Okay.
Yeah.
do you have a question?
I do.
Go ahead.
Hi, it's Greg. I have a question. How controversial is it when you talk to the honeybee people that the honeybee colony is unequal? It's not a bee democracy.
Yeah. So actually, the reason why we wanted to we calculated this Gini coefficient to directly show was to convince our own experimental collaborators, because they they thought that the worker honeybees are identical, and that's honeybee 101. So I think it is still widely assumed that the worker honeybees are identical. Yeah, but we strongly believe our research results shows the other way.
Yeah. Great, thank you. Very nice talk.
Great. Well, yeah, I had a quick question about your hypothesis that the genetic similarity between the bees leads to less inequality. So I'm just curious if you're planning to test this with colonies that have more than one, so one queen, but more than one drone father.
So we are not. So we are looking at like different stuff than this testing this hypothesis, but I think that's something we can do. Yeah.
Okay. Yep. That makes sense.
Yeah.
All right. So that concludes the session. Thank you. Let's thank again to all the speakers. We will follow the instruction by the biology division from APS to see if we can post this video somewhere online, but we will make sure we get everybody's permission first, which will probably happen sometime over the next few days.
I just want to thank everyone for putting up with, somewhat variable conditions and a learning process for us, but I really certainly we all enjoyed hearing from everyone. So thank you for your enthusiasm for today.
Right.
Good.
Okay. Dismissed.
That's less good virtually.
Yeah,